Good evening, everyone. So let's start with week 12. This is the last week, but I will take one more class as a summary class. So I will discuss like all the topic that we have covered on Wednesday. So this is kind of the content last class. So let's start with the content that we will try to cover today. So, today we will cover the lecture 59 to lecture 63, and firstly, we will try to cover different electron acceptor, discuss about the electron acceptor and donor. Then we will come to the application of the microbiology. It can be fermentation, it can be like we can do have some testing in the environmental sciences, for example, BOD test that we can do. Then we can see the biodegradation potential of municipal solid waste. So what kind of bacteria need what kind of like temperature and other things needed for the biodegradation of municipal solid waste. So we will try to understand that. Then we will try to understand municipal wastewater treatment, though we do not have much question on this topic, but we will try to cover this as well. Then we will a little bit talk about the bioleaching of heavy metals. Then we have multiple questions on biogeochemical cycle. So like the difference between the carbon cycle, nitrogen cycle, sulfur cycle, phosphorus cycle, even iron cycle, mercury cycle. So we will try to discuss this through a lot of MCQ question, and I encourage you to answer either using your chat box, or like you can unmute yourself and you can reply me, because I think discussion is the best part that we can do in the live session. Okay, so I start my first question. Here, so this question is, what is the primary function of water molecule during the light dependent reactions of photosynthesis? And we have four options here: to provide oxygen for the Kelvin cycle, B is to donate electron to photosystem 2, C is to act as a substrate for ATP synthase, and D is to capture light energy. So we, I think in previous week, we already have discussed that in the photosynthesis, we do have two type of reaction. The one is light reaction and another is dark reaction. So this question is based on the light reaction. And what is the main purpose of the light reaction? So can you people tell me, out of these four option, which one could be the right one? Even if you are giving me the wrong answer, I will correct you, so don't worry about this. But I think with the communication, with the discussion, you will learn more effectively. So that's why I always encourage you people to speak up, and to you can write your option in the chat box anytime. So what is the primary function of water molecule that we use in light dependent reaction of photosynthesis? Okay, so here the answer will be to donate electron to photosystem 2, and let me discuss this actually. So just see here, this is the chloroplast. Let me use the some pen and the red color. Okay, so just see here, this is the membrane of your chloroplast, and this is your thalcaloid lumen. This is your thalcaloid lumen, and just keep in mind, this is your chloroplast. And in that chloroplast, what will happen? There is a one structure, and that structure is P680. So this is, you can say, the chlorophyll pigment that will take the sunlight at the wavelength 680 nanometer. Okay, so whenever there will be sunlight, here that will strike to that pigment or that molecule, but that particular sunlight should have the wavelength of p6 680 nanometer. Then what will happen, and we do have the water in the chloroplast. So what will happen, this water will be break down into O2 and H plus. So and then what will happen, based on this, electron will be released. So this is important. So whenever there is the splitting of water and the sunlight will strike, electron will be released. So the two thing will happen. The one is electron will be released, and the second is H plus ion will be released. Okay, so this electron will go on through different molecule. This electron is going through different molecule, and again will go whenever, like, we do have another molecule or pigment. That pigment will take the sunlight of the wavelength 700. Okay, so and the finally what will happen, this electron what has been released from here, this reaction, this electron will be used to create NADPH from NADP. Okay, and the hydrogen ions that we had here, H plus ions, these H plus ions will be used to convert ADP to ATP. So only two things that you need to remember. The first thing is there will be splitting of water into O2, H plus ion and electrons. And these electrons will move in this direction, and these electrons will be taken up by NADP, and that will convert this NADP to NADPH. And the another thing is, after this electron consumption, we also have H plus ions that has been released. These H plus ions will be utilized here, where the ADP will be converted into ATP. So two things happened. Using electron, we changed NADP to NADPH. And second thing what we did, we do have H plus ion, and using that H plus ion, we had some gradient, proton gradient, and using that proton gradient, we converted ADP to ATP. So means we have generated energy. We have generated energy, and we utilized H plus ion and electron from the splitting of water using this light reaction. And this is light reaction. This is after that. So this is the light reaction. Afterward we will go to the dark reaction. And what will happen in the dark reaction, because you know that plants take CO2. Plants take CO2. We have generated this NADPH, we have generated ATP. This NADPH ATP will be utilized to convert this CO2 to glucose and water. And water. So this is how plant can synthesize their own food, because we are generating energy whenever there is a splitting of water. So what we are saying, then this water is the electron donor, right? This water is the electron donor, and this O2 is just the side product of the splitting of water. So so our question was, let me show you the question. So our question was, what was the primary function of water molecule during the light reaction of photosynthesis? So it will donate electron to the photosystem too, and then what will be the final thing? That NADP will be changed into NADPH, and ADP will be changed into ATP. So this is the primary use of water molecule during the light dependent reaction of photosynthesis. I think this is clear. If it is not clear, you can raise your hand or you can write in the chat box. I try to explain it. So should we go to the next question? Anyone? If things are clear, should we go to next question? Okay, so I'm going to the next question. Okay, and what I was, let me also explain this. One slide also added here. After this light dependent reaction, there is a dark reaction, and as I told you, the CO2 will be used, and we have created ATP, we have created NADPH in the light reaction. So these will be utilized to create glucose and another sugar. So this is known as the Kelvin cycle. This will again be in chloroplast, but the thing is, here the food of the plant will be synthesized. So this is the next step of the light reaction. So in light reaction, we just released electron from the water, and that were used to change into ATP and NADPH in the dark reaction. So this is the part of dark reaction. So in dark reaction, CO2 will be utilized. We have ATP and NADPH. Using that, we will synthesize the food. So this food will be synthesized by the plants. So just you can see here, in the light dependent reaction, water molecules are split in the process known as photolysis. So in the presence of photons, in the presence of sunlight, water will be splitted. This splitting yield electron, proton, and oxygen. Electrons are donated to photosystem 2 to replace those lost during the excitation by light reaction. This electron donation is crucial for maintaining the flow of electron through photosynthetic electron transport chain. So this is like light reaction and dark reaction. This will complete the process of photosynthesis, right? Okay, coming to the next question. Okay, because I have explained the photosynthesis, the light reaction, and then the dark reaction. I think now you can give me the answer for this. Which of the following statement accurately describe the fate of oxygen produced during the light-dependent reactions? And we have four options. It is used directly in the Kelvin cycle. It combines with NADP to form NADPH. It is released into the atmosphere as a byproduct. And the last option is it acts as an electron donor for photosystem 1. As I told you, in the presence of sunlight, we had the splitting of the water, right? And splitting of water will release oxygen and proton and electron. And I have told you the function of electron, what how this electron will be utilized. I have told you the function of H plus sign, that is the proton, what will be the function of proton. But along with that, oxygen was also released. So what will be the fate of that oxygen that has been produced during the light dependent reaction? Is there any function of this oxygen in whole photosynthesis, or it is just the a byproduct and that will be released into the atmosphere? So can you give me the answer for this? Yogi gave me C answer, and yes, I agree with this. Let me show you. So again, I have the same slide, and just see here. Uh, we had water, and this water has been splitted into H plus ion and electron. So electron, H plus ion. So these are being using. These we are using, but this is just the byproduct, and this byproduct will be released into the atmosphere. And let me just read here the content that I have written. So during photosynthesis, in the light dependent reaction, water molecules are split to provide electron, as we already have discussed, resulting in the formation of oxygen and proton. And this oxygen produced is not utilized in the subsequent step of photosynthesis, but it will diffuse out of the chloroplast. And if it diffuse out from the chloroplast, it will be present in the atmosphere, and it is released into atmosphere as a byproduct. And because this is very important, uh, uh, in sulfur bacteria, in sulfur green bacteria, and in sulfur uh, purple bacteria, you will read that this electron and H plus ion we can produce not only from H2O but from H2S as well. So sulfur bacteria we can also use this H2S. So if we are replacing this water from H2S or from some other molecule, then what will happen? No doubt they will release electron, they will release proton, but they will not release oxygen. And this oxygen is very important for us to have at least 21% in the atmosphere. Otherwise there will be very much difficult in the survival of the human being. So in sulfur bacteria, these sulfur bacteria are also having the photosynthesis, but the point is that oxygen is not being releasing using that photosynthesis. These are just the plants that are utilizing water, but bacteria are utilizing H2S or some another molecule by which they can do the photosynthesis. But this is the green plant that are using water, and these are releasing oxygen as the byproduct, and this oxygen is important for us, right? So the option was that this is just oxygen, whatever has been released in the light dependent reaction is the byproduct. Okay, so the next question is, we already have discussed this one as well using that figure, but still I have this question. And this question is, in the context of photosynthesis, what is the significance of the proton H plus ion released during the splitting of water molecule? And we have four options. They are used to produce NADP to NADPH. Second option is they help establish a proton gradient across the thalcoid membrane. C is they directly participate in the synthesis of glucose. And D is they replace the electron lost by chlorophyll molecule. So we have learned till now that uh, water will be splitted in the presence of sunlight. Electron will be released, proton will be released. This electron will be used to convert NADP to NADPH. And this proton will be used to convert ADP to ATP. But this conversion will be happen only when there is a proton gradient, and this proton gradient will be when there will be more number of proton or H plus ion. So now you can tell me the significance of proton that will be released during the splitting of water molecule. So what do you think? What is the right answer for this? Anyone? What is the use of proton that has been released after splitting of water in the presence of sunlight in the chloroplast of the plant? We already have discussed that it will establish the proton gradient. It will establish, and in the last to last class, we have established the fact that if we have any membrane, if we do have any membrane and we have H plus ion, H plus ion, H plus ion, then what will happen? There is a one molecule, there is a one complex, ATP complex, where this ADP will change into ATP, when this H plus ion will come out or will come in. Whenever we are changing ADP to ATP, there should be a proton gradient. and otherwise the reaction will not happen. So for enzyme ATP synthase, there is a condition that there should be H plus ion gradient, and that whenever your H plus ion out of this gradient, if one H plus ion will come in, then what it will do, ADP will change into ATP. So to establish the proton gradient across the thalcoid membrane. So thalcoid membrane is like inside the chloroplast, we have the thalcoid membrane. And to establish the proton gradient, we will use protons that has been released after the splitting of water molecule. I must have here. So just see here, this is your thalcoid membrane. This is your thalcoid membrane. And we have like after splitting of water, we do have a lot of H plus ion. We do have lot of H plus ion. And what will happen? We have one complex, ATP complex. And in that ATP complex, if this H plus ion will go inside, because there is a gradient has been created where outside, so in the thalcoid interior space we have a lot of H plus ions, and here in the stroma we do not have, so we have created the proton gradient. So in the presence of ATP gradient, this H plus ion will come inside, and this ADP will be converted into ATP. So this is the role of H plus ion that has been released after splitting of water molecule. So here I have written the writer. So the proton released during the photolysis of water contribute to proton gradient across the thalcoid membrane. This gradient create a form of potential energy known as the PMFs, and this PMF terminology is very famous. So this is proton motive force. ATP synthase utilize this force to derive the synthesis of ATP from ADP. And in organic phosphate, this process called chemoosmosis is essential for producing the ATP required in the Calvin cycle for the synthesis of glucose. So I think this is clear that we had water. Water will release electron, water will release proton. This electron will be used to change NADP to NADPH, and this proton will be used to convert ADP to ATP. And this NADPH and ATP will be used further in the Calvin cycle, so that using CO2 we can synthesize glucose in the plant, and this whole process is known as photosynthesis, right? Okay, so coming to the next question, what is the primary role of photosystem 2 in non-cyclic photophosphorylation? And we have four options: to reduce NADP to NADPH. The second option is to absorb light energy and initiate the splitting of water molecule. C is to facilitate the fixing of CO2. D option is to transfer electron back to photosystem 1. So we have talked about photosystem 1 and photosystem 2, right? Let me show you again. So this is our photosystem 1, and this is our photosystem 2. And in this photosystem 1 and 2, in both, we will utilize sunlight. We are utilizing sunlight, but at a particular wavelength. Okay, so this is a 700 this nanometer wavelength of sunlight we are utilizing here. 680 nanometer sunlight we are using. So we are using different wavelength of sunlight here in photosystem 1 and 2. We have learned it, right? And there is another term. So you people right now know the what do you mean by a photosystem 2, right? The next is non-cyclic photophosphorylation. So photo means light, phosphorylation means if you have ADP. So ADP is adenosine diphosphate. So we have two phosphate, but in the presence of H plus ion and in the presence of one phosphate, we can convert into ATP. So in the presence of sunlight, in the presence of phosphorus, what we can do, we can do the process that is photophosphorylation. So this photophosphorylation is in the presence of sunlight, adding the phosphate, one more phosphate into ADP, that will change ADP to ATP. So this is photophosphorylation. So we have learned what do you mean by photosystem 2? We have learned what do you mean by photophosphorylation. And another is non-cyclic. So non-cyclic means, as we already have discussed, that the splitting of water will release your electron. So so there is a two condition, that this electron will be recycled or not. So if it is being recycled, then we will tell this is no this is cyclic. And if these electrons are not recycled, or then we will call it non-cyclic. So here the question is, what is the primary role of photosystem 2 in non-cyclic photophosphorylation? So I have explained much about the terminology that we have in this question. So now you can give me the answer for this. What do you think? What is the right answer for this question? Anyone want to give me the answer? Okay, so the answer is the primary role of photosystem 2. We just need to tell the role of photosystem 2. So and as I told you, photosystem 2 means there is a complex that will absorb the light, but at a particular wavelength. So the role was to absorb light energy and to initiate the splitting of water molecule. So this is the primary role of photosystem 2. We already have learned from question 1 to till now, we just learned that much only. And just see here, and I wanted to tell you about the cyclic photophosphorylation and non-cyclic photophosphorylation. So we have learned, we had the chlorophyll, right? We have the photosystem 2, right? We have the photosystem 2, then electrons are released after splitting of water, and these electrons will be used so that these electrons will be used so that we can change an ADP to an ADPH. But if we are if but if we are not utilizing this electron, then this electron will go back to the chlorophyll only. So it means what we are doing, this system is not changing an ADP to an ADPH. So just see here, there is only one photosystem that will involve. This is the photosystem 1, and this photosystem 1 activation center is P700. So means we will utilize the wavelength of sunlight, that is 700 nanometer. Here the electron travel in the cyclic manner, then this electron travel back to the photosystem 1. But in non-cyclic, in most of the green plant, the photophosphorylation is non-cyclic in nature, because this electron will be utilized to convert an ADP to an ADPH. And here, and that's why here, because we are not utilizing the electron, so that's why only ATP will be produced. This cyclic photophosphorylation will not release an ADPH, because we are not utilizing the electron for it. Then photolysis or water splitting is absent, because whenever there is the water splitting, that will release the electron. But if we are using one electron always, so it means there is a no splitting of water. Oxygen is also not involved. If we are not splitting the water, then from where this oxygen will come? So oxygen is also not involved, and this system is prominent in bacteria. So this is cyclic photophosphorylation, and this is the light reaction part of the photosynthesis. We are not talking about the dark reaction of the photosynthesis where CO2 will be utilized to produce the glucose. Okay, so this is the cyclic one, and let me show you the non-cyclic one as well. This is very common in the green plant. So we have for chlorophyll, then we have water, and it is releasing electron and H plus ion. This electron will be utilized to change an ADP to an ADPH, and this H plus ion will create the proton gradient, and using that proton gradient, we can synthesize ATP. So if we do have the non-cyclic photophosphorylation, if we do have this kind of reaction, then we have both an ADPH and ATP that will be produced. And this is very common in the green plant. And here we do have both photosystem 1 and 2. Activation reaction center is P680. So the sunlight wavelength will be 680 nanometer. Here the electron travel in non-cyclic manner. We already have learned this. And electron from photosystem 1 is accepted by an ADP. We already have learned here. And then there is the splitting of water, and oxygen will involved as a byproduct. And already we have talked another thing. So just I'm reading the write up so that everything is clear in your mind. So photosystem 2 is the first protein complex in the light dependent reaction of the photosynthesis. It absorbs light energy, which will excite the electron to the higher energy state, and then high energy electron will be transferred in the electron transfer chain. And to replace this lost electron, photosystem 2 facilitate the photolysis of water molecule, resulting in the release of electron, proton, and oxygen. We already have learned, and this will come under non-cyclic photophosphorylation. So I think it is now clear what do you mean by cyclic photophosphorylation and what do you mean by non-cyclic photophosphorylation, and what is the role of photophosphorylation, and what is the role of photosystem 1 and photosystem 2. We have learned everything, right? If there is any doubt, you people can tell, otherwise we can go ahead with the next question. Is everything clear till now? Anyone can write in the chat box if everything is clear, or if there is any doubt, then also I can like explain more about something if something is not clear. Okay, so I'm going to the next question. Okay, we know all the terminologies, right? Non-cyclic phosphorylation, electrons, photosystem 1, and we have seen what was the mechanism. So I think this is again a very familiar question to you. Of this question, you can answer. So this question is: during non-cyclic photophosphorylation, the electron excited in photosystem 1 are ultimately used to, and we have four options. It will generate a proton gradient for ATP synthesis. It will reduce an ADPH to an ADPH. It will replace electron lost in photosystem 2. It will split water molecule into oxygen and protons. So the question is, these electrons that has been released, how they will be utilized? So the question is, are ultimately used to. So for what purpose, for what is the function of that electron that we have that has been excited in photosystem 1? What is the use of these electrons? We already have learned, right? So these electrons, like we have learned in non-cyclic phosphorylation, uh, an ADP will convert into an ADPH, but for that conversion, electron, and these electrons will be come from the splitting of water molecule. Right? This is very clear. So I don't think that we need much explanation, and we already have discussed this non-cyclic photophosphorylation. Water will be used, electron will be released, H plus ion will be released. These electrons will come through a transport chain, and ultimately an ADP will be changed into an ATP if we do have enough electrons, and this H plus ion will be used to synthesize the ATP. So this was the ultimate aim of releasing the electrons from photosystem 1 or photosystem 2. Okay, so coming to the next question. Okay, like from the figure, from this schematic figure and mechanism, we try to understand the light reaction. And now ultimately the question is, you need to tell me the sequence that will correctly represent the flow of electron during non-cyclic photophosphorylation. And options are photosystem 1 to photosystem 2 to an ADP. B option is photosystem 2 to photosystem 1 to an ADP. C is photosystem 2 to an ADP to photosystem 1. And D option is photosystem 1 to an ADP to photosystem 2. So can you tell me the flow of electron that we had during non-cyclic photophosphorylation? Till now we have one figure where I showed that how this electron will move, and ultimately what is the use of that electron. So can you tell me the correct sequence of the flow of electron that we have learned during non-cyclic photophosphorylation? Anyone? This is very simple question. If you people are actually like watching this video, and we had we try to understand the light reaction, then you must know the answer for this. Can you tell me, or should I tell it? Okay, so the answer is, firstly we had photosystem 2. The water splitting will happen in photosystem 2. Then this the release of electron will go to photosystem 1, where there will be the excitation of this electron, and then this electron will be ultimately used to convert an ADP to an ADPH. So this is the right sequence: photosystem 2 to photosystem 1 to an ADPH. Ultimately, electron will be used. So in non-photosphorylation, non-cyclic photophosphorylation, electron flow in a linear path. This process begins with photosystem 2, where light energy excites electron that are then passed through the ETC, that is the electron transport chain, to photosystem 1. In photosystem 1, the electrons are re-energized by light absorption and eventually used to reduce an ADP to an ADPH. And this linear flow of electron is essential for the production of both ATP and an ADPH during the light dependent reaction of photosynthesis. We have like learned much about this fact. Okay, so coming to my next question. As I told you, there is a splitting of water in green plants, but it is not always possible that we have water presence and this green sulfur bacterias are not just let me show you. We have learned that we had H2O. This H2O will be splitted into O2 plus H plus ion and plus electrons. These electrons will be used for the synthesis of NADP, NADPH, and we have learnt that this H plus ion will be utilized for the conversion to ATP. And this is just the byproduct, and this byproduct will released into the atmosphere. So this water is nothing but this is electron donor. This is nothing but the electron donor. In green plant, water is the electron donor. But if we have green sulfur bacteria, and where we do not have like here, the photosynthesis is not oxygenic, but here the another kind of photosynthesis is anoxygenic photosynthesis. So what in spite of this, so this will not be here. So what kind of molecule will be utilized in green sulfur bacteria so that there can be the photosynthesis, but that photosynthesis will be anoxygenic. So the question is, which of the following is the primary electron donor utilized by green sulfur bacteria during anoxygenic photosynthesis? And we have four options: water, then hydrogen sulfide, then oxygen, and carbon dioxide. So what do you think? What is the right answer? So if we do not have water as electron donor, so what molecule we can use as the electron donor in green sulfur bacteria? So we have learned in green plant we are utilizing H2O as electron donor because it is producing electrons, right? But if we do not have water presence, and if we do have green sulfur bacteria in place of green plant, then what will be the electron donor for that green sulfur bacterias? Any guess about this? So this will be hydrogen sulfide. This will be hydrogen sulfide. So here this is H2S. So if we do have H2S, then this H2S no doubt will release hydrogen a proton. This will release electron, and this will again act as the like electron donor. But along with that, this is not releasing oxygen, but this is releasing sulfur. So there is no doubt photosynthesis. The bacteria will produce their food by their self. There is a photosynthesis, but that photosynthesis is not oxygenic. So that photosynthesis will not produce oxygen. So that's why this photosynthesis is anoxygenic, because we are releasing sulfur. Okay, so that's why whatever the photosynthesis is happening in the green sulfur bacteria, this is not oxygenic, but this is anoxygenic, because the splitting of H2S is not releasing the oxygen as a byproduct, but this is sulfur, that is the byproduct after splitting of H2S, because as electron donor we are utilizing H2S in the place of water. So I think this is now clear what will be the electron donor for the green sulfur bacteria. And let me show you here. So the ultimately, obviously, like in green plant as well, CO2 will be utilized so that we can produce the glucose. But in the green plant, we are using H2O, but here in green sulfur bacteria, we are utilizing H2S and light. So in place of oxygen, in green sulfur bacteria there is a sulfur that will be produced, and along with water and glucose. So just see here, uh, initially H2S will be taken by the sulfur bacteria. Sulfur will be released in the atmosphere, and because using that electron donor, we had proton, so no doubt ATP and NADPH will be generated, and this will be utilized when CO2 will come and we can produce glucose. So everything is same, but in place of H2O we are utilizing H2S, and because of that the byproduct is not oxygen but this is sulfur. So just see the write-up. Green sulfur bacteria, such as those in family Chlorobiaceae, perform anoxygenic photosynthesis by utilizing hydrogen sulfide as an electron donor, unlike oxygenic photosynthesis, which uses water and release oxygen. And these bacteria oxidize H2S to elemental sulfur or sulfate without producing oxygen, and this adaptation allows them to thrive in anaerobic environment rich in sulfide. So for example, if there is any coal mining or where there is a rich sulfide, then at that place bacteria can do the photosynthesis, bacteria can survive there because they can utilize H2S in place of H2O as electron donor. So is it clear, everything? And because there was the term oxygenic photosynthesis and anoxygenic photosynthesis, so just see here, if thank you Ganendra for the response. So if we do have oxygenic photosynthesis, so this can be happen in cyanobacteria and eukaryotic phototrophs. But if this is the anoxygenic photosynthesis, then we have purple and green sulfur bacteria and even non-sulfur bacteria. And we already have discussed, in the place of H2O as an electron donor, in the green and purple sulfur bacteria we will utilize mostly H2S. And here we do have aerobic zone of lake, river, or ocean where there will be photosynthesis. But in anoxygenic photosynthesis there is the anoxic condition, means there is no presence of oxygen. So just keep in mind, bacteria are also doing the photosynthesis, but in that photosynthesis oxygen is not being released because they are not utilizing water for the electron donor, they are utilizing H2S as the electron donor, and because of that the byproduct is elemental sulfur but not the oxygen. And this is the difference between the photosynthesis between bacteria and the green plants. Okay, so coming to the next question, in an exogenic photosynthesis, why is water not used as the electron donor by green and purple bacteria? So why they are not using water? Why they are using hydrogen sulfide as the electron donor? So we have four options here. Because water is toxic to these bacteria. B option is because they lack the necessary enzyme to oxidize water. C is because water do not provide sufficient energy, and D is because water inhibits their photosynthetic pigments. What do you think? Why these bacteria are not utilizing H2O, but they are utilizing the hydrogen sulfide? So just imagine, we have like billions of bacteria, and if these bacteria, can you mute yourself, Ravi? Okay, thank you so much. So just imagine that we have billions of bacteria, and if these billions of bacteria are giving us oxygen as the byproduct, then we will like environment will be enriched with the oxygen concentration, but this is not happening because whatever the photosynthesis is happening in the uh, green and the sulfur bacteria, this photosynthesis is not giving us the byproduct oxygen. So why this is happening? Why they are not utilizing the water as the electron donor, but they are utilizing hydrogen sulfide as the electron donor? What is the limitations of this bacteria that they have? So because they lack necessary enzyme to oxidize water. As I told you, each and every reaction that is happening in the plants, in the human, every reaction is dependent on particular type of enzyme. That particular type of enzyme uh, will catalyze that reaction, and if we do not have that particular enzyme to catalyze that reaction, then that reaction cannot be happen spontaneously in the nature. So because these bacteria do not have necessary enzyme that can oxidize this water, and because of that, they are not utilizing water, but they are utilizing H2S, that is the hydrogen sulfide, as the electron donor. So that's why B answer is the right answer. Just see here the explanation. Green and purple bacteria do not use water as an electron donor because they lack the complex enzymatic machinery such as oxygen evolving complex found in cyanobacteria and plants required to oxidize water and release oxygen. Instead, they utilize their electron donor like hydrogen sulfide, hydrogen gas, ferrous iron to derive their photosynthetic processes. So I think this is clear now. And let me move to the next question. Which of the following best describe the environmental condition where purple sulfur bacteria are commonly found? And four options are here. Aerobic environment with high light intensity. B is anaerobic environment rich in hydrogen sulfide. C is dry oxygen rich terrestrial soil. And D is deep oceanic zone devoid of light. So you have to tell me uh, what is the criteria, what is the environmental condition where this purple sulfur bacteria can survive, can thrive. And we already have discussed, right, that this sulfur uh, purple sulfur bacteria or green sulfur bacteria are doing the photosynthesis. That photosynthesis is not oxygenic but anoxygenic. So means they are not releasing oxygen, but and they are not splitting of water, but they are using hydrogen sulfide for the electron as a electron donor. So based on this explanation, I will go to the B option, that is anaerobic environment. So there is not the presence of oxygen because oxygen is not being produced by as the byproduct. So there is a no oxygen there. And because we are splitting here H2S, and this H2S will release proton, electron, and sulfur, but they are not releasing water, sorry, oxygen. And that's why there is an anaerobic environment, and the environment where there is a lot of electron donor compound, and that electron donor compound is hydrogen sulfide, but this is not water. So that's why B answer is the correct one. So just see here the explanation. So such kind of bacteria are typically found in anaerobic environment where there is a abundant amount of hydrogen sulfide, such as stagnant water bodies, sulfur springs, and illuminated anoxic zone of light. So where there is a no oxygen, and they perform anoxygenic photosynthesis using H2S as an electron donor, oxidizing it to elemental sulfur or sulfate, and this condition is ideal for the metabolic process and the ecological niche. Okay, so next coming to the another question, and this question is, we already have learned, right? We do have the one pigment that is the chlorophyll, and that chlorophyll is utilized for the photosynthesis, right? But we do have various type of pigment in the chloroplast or in the plants, and you have to tell me about the pigment. That pigment can act as an accessory light harvesting molecule and also protect chlorophyll from photooxidative damage. And this pigment may be out of these four options, and you have to tell me the one option. And these options are anthocyanin, phycobilin, then carotenoids, and the next is fucoxanthin. So we have learned that we have chlorophyll A, chlorophyll B, chlorophyll C, and that chlorophyll will harvest, will utilize the sunlight, and then the splitting of water will happen, electron will be released, proton will release, and that electron and proton will use further. But along with this chlorophyll, we have another pigment, and that pigment will not only enhance the utilization of light, but this pigment will also protect chlorophyll from oxidative damage. So can you tell the name of that particular pigment that we are talking about? So this pigment is carotenoid. And let me show you the explanation of this. So just see here, we have chlorophyll, right? We have chlorophyll A, chlorophyll B, and I told you we have the peak of this. Just see the peak of this, and so this is near to 480, right? Near to 480 nanometer. So it means there is the reflection of the green light by the chlorophyll B, and it means, and but here this zone, just see the this zone, in this zone there is the absorbance of light by this chlorophyll B. But it may be that like utilizing that particular wavelength is very difficult. It may be because light have a very long wavelength range. So we do have another pigment, for example carotenoid, we have xanthophylls. So these pigment can enhance the absorption of sunlight, they can enhance or they can broaden the wavelength range of which the sunlight can be utilized. Okay, so the function of this one is to enhance the light harvesting system, right? And the second is it can also protect the chlorophyll from the photooxidative damage. So just see here, carotenoids serve dual function in photosynthetic organism. They act as accessory pigment by absorbing light in blue green region of the spectrum and transferring energy to chlorophyll, and therefore they are expanding the range of light that we can use for the photosynthesis. And along with that, it can also protect chlorophyll from photooxidative damage using two type of processes. And the one like major process is the quenching ROS, that is the reactive oxygen species, and this is how it will dissipate that excessive energy as heat, and this is how it can prevent the formation of harmful singlet oxygen. So there is a two function of carotenoid. The first is it will broaden the wavelength of the light that plant can absorb for the photosynthesis, and second is it will protect the chlorophyll from the oxidative photooxidative damage. Okay, so based on that only, we have another question, and that question is, what is the primary mechanism by which carotenoid prevent photo damage to photosynthetic apparatus? So we have learned that carotenoids will prevent the chlorophyll from photooxidative damage. But what is that process? By that process, it can prevent this photo damage to the photosynthetic apparatus. So the answer is that it will quench chlorophyll triplet state or singlet state. Let me show you here. So C is the right answer, and I think I have one figure by which you can see. So just see here, this is the carotenoid, and if we do have light energy, if we have beta carotenoid, we have carotene, then we, if we do have light energy, then this will do the physical quenching. This will do the physical quenching, and based on that, uh, it can prevent the chlorophyll uh, uh, being uh, uh, being getting oxidized, right? So just see here the explanation. Carotenoid protect the photosynthetic apparatus by quenching the excited triplet state of chlorophyll and singlet oxygen, both of which can lead to the generation of reactive oxygen species that can cause the oxidative damage. And this quenching mechanism will involve the transfer of excessive energy from chlorophyll to carotenoid, which then dissipate it as a heat. Thereby, this is safeguarding the photosynthetic machinery. Okay, I think I should go now to the next question. Again, this is the same figure, and that how this is like it will generate the stress signaling, and how carotenoids will be used for the quenching, and this is how it can prevent the chlorophyll getting oxidized and getting damaged from sunlight. Okay, the next question is, if we do not have the carotenoid, then what would happen to the plant that has been exposed to high light intensity? The like very similar question to the previous one, but in a different form. And we have four options. They would photosynthesize more efficiently. B option is they would accumulate excess chlorophyll. C option is they would suffer photo damage leading to impaired photosynthesis. And D is they would become more drought resistant. So if we do not have carotenoid, we have learned the function of carotenoid. If there is no carotenoid, then what will happen if the plant is exposed to high light intensity? If you people know that if there is enough ultraviolet radiation in the sunlight, then there is a genetic mutation in the human being, and there may be the cancer. But what is the we do have the carotenoid, right? And this carotenoid can help the plants to to reduce this photo oxidation damage. And because photosynthesis sorry the sunlight that have the high light anti intensity can be very harmful to the plants. So what will happen if this is the happening, if we do not have carotenoid in the plants, and if the plant is getting exposed to the high light intensity? We already have seen the function of carotenoid, right? So based on that, I think you can give me the answer for this question. So this is, if we do not have the carotenoid, then we will suffer, the plant will suffer from photo damage, and this will lead to the impaired photosynthesis. Just like human, if we do have the ultraviolet radiation in the sunlight, then we will have the cancer, there will be the genetic mutation. Likewise, but we have like some kind of protection that we have in our human, and if that protection is not sufficient, then we are using the sunscreen, right? But in the plants cannot use sunscreen, right? And they have to protect themselves from this high light intensity, so they have the pigment, and that pigment is carotenoid. That pigment can prevent any damage that can be caused from that can be caused by the sunlight that have the high light energy. And if the carotenoid are not present, then this will lead to the impaired photosynthesis. Okay, so coming to the next question, I think we already have explained much about this, so we can skip this part. Okay, coming to the next topic. So till now we talked about photosynthesis, we talked about the chlorophyll, we talked about anoxygenic and oxygenic photosynthesis. Now coming to the application of microbiology, and in like we do have different type of bacteria, but out of this different variety of bacteria, we have two bacteria that are methanogens and methanotropes. And you need to tell me the difference between these two type of bacteria. The one is methanogen and the different another is methanotrop, because as we right now we are talking about the application of microbiology. And if you have learned that we can produce the methane, or if there is a methane environment, then we have different type of bacteria that can be methane environment. So, and if methane is produced by some bacteria, then we can use that methane as a fuel in our transport system. And like this is the application of different kind of bacteria. So okay, so coming to the options, you just need to tell me the difference between methanogens and methanotropes. So the four options are: methanogens utilize methane as a carbon source, while methanotrope produce methane. B option is methanogen are aerobic bacteria, whereas methanotropes are anaerobic archaea. C is methanogen produce methane under anaerobic condition. Methanotropes consume methane under aerobic conditions. And D is methanogen and methanotrope both produce methane under aerobic condition. You just have to tell me the difference between this. I think in municipal sewage treatment, you must have learned that if we do have organic matter, and this to degrade that organic matter, we can use aerobic bacteria, we can use anaerobic bacteria. And if we are using anaerobic bacteria, then CO2 is not releasing. This is the methane that is releasing, and we can utilize, we can channel this methane to the pipes, and we can use it for the fuel. But the so this is just the application of microbiology. But before that, you must know, no, what kind of bacteria you can use for that kind of reactions. And again, I will come to the question that we have out of a lot of variety of bacteria, we have two bacteria, methanogens and methanotrope, and you have to tell me the difference between these two. So the answer is, if the bacteria is known as methanogen, then this will produce methane under anaerobic condition. Okay, this is producing methane, right? But if we do have methanotrope, then this will consume the methane. The one is producing methane and another is consuming methane. And if this is producing methane, this will be under anaerobic condition. But if it is consuming methane, it is not always the very mandatory statement. Then there should be anaerobic, but there may be aerobic condition uh, if some bacteria are consuming methane. So C is the right answer for this question. And I have one diagram as well. So we you can see methanogen versus methanotropes. So under methanogens, any methane producing bacteria, especially archaea, that reduce carbon dioxide to methane. And methanotropes are any group of aerobic bacteria that are capable of utilizing methane as the carbon and the energy source. So they are producing methane and they are utilizing methane. And it will be under anaerobic condition, and this will be under aerobic condition. And this will produce methane, and methane is the greenhouse gas. And this will reduce the effect of methane because they are consuming the methane, right? I think that I should not go to the explanation because we have learned the difference between these two. And here again, you can see if we do have methanogens, then this will release the methane. And this methane will be utilized by the methanotropes and CO2 will be released. So just see here, CO2 will be utilized here, but a methane will produce, and this methane will taken up by methanotrope and CO2 will be released. So just see the beauty of the nature. One is taking CO2, releasing methane. The another bacteria is taking methane and releasing CO2. So this is the kind of balance that we have in the nature. Okay, so let me come to the next question. The next question is, which form of mercury is considered the most toxic to human? This is very general question that are being asked in net exam. Which mercury is more toxic to the human body? So we have four options: elemental mercury, mercuric chloride, methyl mercury, and mercurous chloride. Very simple and very straightforward question every time asked in the net environmental science paper, like which mercury is more toxic to human. Any guess about this? Which mercury form is most toxic to human? So the answer is methyl mercury. So methyl mercury is CH3 and mercury Hg plus. And just see here, we may have different type of mercury that can be present in the atmosphere. It may be elemental, where the oxidative state of that mercury is zero. We can have the organic, such as methyl mercury and ethyl mercury. We can also have inorganic mercury, for example, mercury chloride. We also have the metallic mercury. So if some metal is attached to that mercury, we may also have suspended mercury, for example, mercury sulfide. But if we are talking about the most toxic form, then uh, just see here, if we do have elemental mercury, that will be in liquid in nature, and that is it is toxic in nature, no doubt. And this mercury will be used into in thermometer, in light bulb, and even in dental amalgams. But if we do have inorganic mercury, this will be in mineral form, and like a lot of material can be synthesized using that inorganic mercury, for example, and for different processes, for example, tanning, dyeing, and pharmaceuticals. But if we do have the organic mercury, for example, methyl mercury, this is in vapor form, right? This is very important. Methyl mercury is in vapor form, and this is very toxic because this vapor we can inhale, right? And this methyl mercury we are using in disinfectant, in fungicide, in wood preservatives. And also this methyl mercury is also present in the fish, and that is the only reason that we are having the smell that we have in beaches or in the area where we are selling the fish. So this is methyl mercury. So I'm just reading the content written here. So methyl mercury is an organic compound of mercury that is highly toxic. It readily bioaccumulates. So why this is very toxic? Because it can bioaccumulate. It can be accumulated in the organism, and it can biomagnifies up the food chain. So whenever we are changing the trophic level, then this is there is the concentration is increased, leading to the significant human health risk in human, including neurological and development impairments. So just keep in mind, methyl mercury is vapor in nature, and this is highly toxic among all the different form of mercury. So right now we are just trying to see the application of microbiology and like how we can degrade this methyl mercury, let's say. Okay, the next question is, in the context of microbial metabolism, which of the following statement is true? And options are, all methanogens are bacteria. A B option is methanotrope produce methane in anaerobic condition. D is methanogens are archaea that produce methane under anaerobic condition. And D is methanotropes are archaea that consume methane under anaerobic condition. So we have learned the difference between methanogens and methanotropes, right? And based on that table that I have provided, you can give me the answer for this. This is about the microbial metabolism. So what they are consuming, what they are releasing. So can you tell me the answer for this? Anyone? Okay. So we have learned, right? Methanogens will produce methane. Methanotropes will utilize methane. And if they are producing methane, so there must be anaerobic condition, right? So methanogens are archaea. These are archaea bacteria, and there, like in a previous week, we already discussed between archaebacteria and eubacteria. So archaebacteria are the bacteria that can thrive in very extreme condition, right? And if the condition is not extreme, then there will be like abundance of eubacteria. So methanogens are archaebacteria because methanogen will be present in very extreme condition. That's why these are the part of archaebacteria, and these methanogens will produce methane under anaerobic condition. And just see here the explanation. Methanogens are a group of archaea known as producing methane as a metabolic byproduct in strictly anaerobic environment. They play a vital role in carbon cycling and are distinct from methanotropes, which are bacteria that will consume methane under aerobic conditions. So we already have discussed the difference between these two, right? Methanogens and methanotropes. So coming to the next question, and right now we are slowly moving to the biogeochemical cycles, the nitrogen cycle, carbon cycle, sulfur cycle, how the one cycle is different from the another cycle. So we are slowly moving to the cycles, biogeochemical cycle. The question is, which of the following process is correctly paired with its associated biochemical pathway? And we have four options. Nitrification, this is under anaerobic respiration. Denitrification process under fermentation. Ammonification under aerobic and anaerobic respiration. And acetogenesis under aerobic respiration. You need to correct the correct pair. We will discuss this as well, but just see here, if it is the ammonification. So ammonification is, we do have nitrogen, um, nitrogen as N2 in the atmosphere, and if we are fixing that N2 into NH3, this is ammonia, then this can be under aerobic as well as under anaerobic respiration, right? So just see here, the first is nitrification. So nitrification is, if we have ammonia, and ammonia is NH3, right? If we do have ammonia, this is NH3, and if we are converting this NH3 to NO2 or NO3, NO3. So this process is nitrification. So, and if we are adding, like it, here it was only water, sorry, hydrogen, but here it is oxygen. It means we are having the reaction with the oxygen here, right? So this can't be anaerobic. This must be aerobic respiration in the nitrification, right? So this is not the right statement. If it is the denitrification, so denitrification is, we have NO2, NO3, and again we are changing into N2. So this is denitrification, and this denitrification is doesn't come under fermentation. So this is again the wrong statement. And this acetogenesis happen if we do not have enough oxygen during the respiration, and that's why this statement is also not correct. So that's why C is only the right answer that we have among all options. So just see here, this is just to tell you what is happening in the nitrogen cycle. So start from here. So this is N2 that we have in the atmosphere. In the atmosphere, using some bacteria, this N2 will be and will be convert into NH3, and this is ammonification. This is ammonification because we are converting this N2 to NH3. Then we have again some bacteria, different type of bacteria, that will change this NH3, that is ammonia, to nitrite, and then nitrite to nitrate, and this process is nitrification. This process is nitrification. And then again, because why we are changing this, because this nitrate is consumed by the plants. This nitrate is consumed, plants cannot consume directly this N2 from the atmosphere, but this should be in the presence of NH3 that plant can utilize. So that's why this NO3 will be utilized by the plant. After that, this plant will be consumed by animal. Then animal, using ammonification, will release NH4 ammonia, and again it will come under NH2. And if we are changing this, we again have some bacteria, that bacteria can convert this nitrate into N2, and this process is denitrification. So just see, we started with the atmospheric nitrogen, that is N2. That N2 changed into NH3 to and NO2 to NO3, because this NO3 is used by the plants. And then again there will be kind of circle. So what I was about to tell is, this is the nitrogen cycle, and in that nitrogen cycle we have this ammonification, and 2 to NH3, this process will come under ammonification. So it involves the conversion of organic nitrogenous matter into ammonium. So here, so like this part, will come under ammonification, and it can occur under both aerobic and anaerobic condition. And various organisms derive energy by oxidizing organic nitrogen compound in diverse environment. Okay, so coming to the next question, which process is primarily carried out by anaerobic bacteria and results in the production of acetate? And we have four options here: nitrification, acetogenesis, denitrification, and the fourth option is nitrogen fixation. So we have two conditions. The first is the bacteria should be anaerobic. So it means it is not utilizing oxygen. And the second condition is that bacteria should produce acetate. So you have to tell me the process where there is anaerobic bacteria and the output is acetate. I think from question only you can guess something. Can you guess the right answer? Can you write in the chat box the answer, whatever you think suitable? Okay, so this is acetogenesis. This is acetogenesis because this nitrification, denitrification, fixation, this is related to nitrogen cycle, right? But if we are talking about the acetate, then acetate doesn't have nitrogen. Acetate have carbon, water, carbon, hydrogen, and oxygen. So this is organic compound and that have CHO. And so if there is the production of acetate, means if there is a compound that have CHO, it means it is we do not have nitrogen, right? Then this nitrification, denitrification, and nitrogen fixation should not be among the right answers. The only answer that we have is acetogenesis. And from terminology also you can guess production of acetate means there must be the process, that is the acetogenesis. Genesis means formation. Aceto came from acetate. So whenever there is the formation or the generation of acetate, then this will be acetogenesis. And let me show you if I have the figure where this acetate can be produced. Yes, so just see here, if we have CO2, if we have CO2 and this CO2 is changed into methane, this is methanogenesis. But if we do have CO2 and this CO2 is generating acetate, then this is acetogenesis. So acetogenesis is the process through which acetate is produced, typically by the reduction of CO2 or organic acid. This process is carried out by anaerobic bacteria, and these bacteria are known as acetogens, which operate under oxygen-free environment, often via fermentation pathway. Okay, so I'm going to next question. If you do have any problem, you can write in the chat box. Otherwise, we will go on to the next question. Okay, so the next question is regarding the nitrogen cycle again. And the question is, in the nitrogen cycle, which process involves the aerobic oxidation of ammonia to nitrate? We have learned, right, in a nitrogen cycle, initially we have nitrogen in the atmosphere in the presence of N2. Then this N2 will be changed into NH3, ammonia. Again, this NH3 will be changed into NO2. This is nitrite. This NH2 will be converted into NO3. Sorry, NO2 will be converted into NO3, and this NO3 is nitrate. So N2 to ammonia to nitrite to nitrate, and again to N2. So this is the nitrogen cycle. And you have to tell me the process where there is the formation of nitrate from ammonia. So can you tell me the name of that process where there is the aerobic oxidation of ammonia, because ammonia is NH3, right? Or ammonium ion NH4 plus, and we do not have oxygen here. But if we are changing it to NO2 or NO3 minus, then there must be oxidation. So can you tell me the process? So this process is nitrification. Let me show you here this cycle. So again, we have N2. This N2 will be changed into a different form, and the one form is ammonia. So just see here, and this is known as the ammonification. This is known as ammonification. And then from this ammonia we will change it into nitrification. We have nitrifying bacteria, and then again some nitrifying bacteria will change into nitrate, and then this nitrate will be taken up by the plants. And we do have denitrifying bacteria that can again change this NO3 to N2. So, and just see here, ammonia to nitrite to nitrate, we have the process. That process is nitrification. So just see the write-up. Nitrification is an aerobic process performed by specific group of autotrophic bacteria and archaea. It involves the oxidation of ammonia to nitrite and subsequently to nitrate. And this process is crucial for the nitrogen cycle in soil and aquatic environment. So I think this is clear now. If we are changing ammonium or ammonia to nitrite and nitrate, this process is known as nitrification. Okay, so I'm going to the next question. Which process allows plants to incorporate atmospheric CO2 into organic molecular? Like the most simplest question ever asked in this class is this one. Whenever we have atmospheric CO2 and plant is doing something so that the CO2 can be changed into organic molecule, so what kind of process we are talking about? So we have four options: respiration, photosynthesis, combustion, decomposition. Plants are taking CO2, plants are synthesize something in in organic molecule, and this process is what? Very simplest, like the simplest question ever asked in this question. So this is photosynthesis, right? In the presence of sunlight, CO2 will be converted into glucose and water and oxygen. So this is photosynthesis. Respiration is opposite to that. We will take oxygen, we will break down the glucose that has been uh, formed by the photosynthesis, and CO2 will be released into the respiration. Combustion is different. Here again, no doubt we are using the CO2, sorry, we are using oxygen, and then CO2 is produced. And decomposition is a different mechanism, different process. And but the explanation that we have CO2 in the atmosphere, plant will utilize it to synthesize some organic molecule. This process is very simply the photosynthesis process. I don't think that I should go into detail because this is very basic question. Okay, again we have very basic question. In the carbon cycle, which of the following serve as the primary carbon source for terrestrial plants? We have four options: carbonate rocks, atmospheric CO2, fossil fuel, and soil organic matter. Because plants need the carbon source because they need to produce glucose, right? So that they have to synthesize their food. So what is the primary carbon source for such kind of plants? Very simple again. This is atmospheric CO2, because we talked about photosynthesis, right? It will take CO2, then water, and in the presence of sunlight, this will this will synthesize glucose, and this glucose will be utilized for the plant for different type of metabolism, right? So this carbon dioxide is the carbon source for the plants. So I don't think that again I should go to the explanation because that was very simple question. Okay, so the next question is about the difference between phosphorus cycle and carbon cycle, and the question is, which of the following best explain why the phosphorus cycle is considered a sedimentary cycle, unlike the carbon cycle? And we have four options here. Phosphorus is primarily stored in the atmosphere. B is phosphorus cycle rapidly. Phosphorus cycles rapidly between living organism and the atmosphere. C is phosphorus lacks a significant gaseous phase and mainly stored in rocks and sediments. And D is phosphorus is abundant in the atmosphere, facilitating its rapid recycling. So can you tell me the difference between phosphorus cycle and the carbon cycle, and why uh, phosphorus cycle is considered as a sedimentary cycle and the carbon cycle is considered as the gaseous cycle? I gave you the hint. The phosphorus cycle is considered as the sedimentary cycle, but carbon cycle is considered as the gaseous cycle. So now can you tell the difference between these two? Why the phosphorus cycle is considered as sedimentary cycle? Because phosphorus lack a significant gaseous phase and mainly stored into rocks and sediments. And because it is stored into rocks and sediments, that's why it is sedimentary cycle. Because most of the part of the phosphorus is present only in rocks and sediments. So that's why this is known as the sedimentary. But if you are talking about the carbon cycle, mostly the carbon is present in the atmosphere in the presence of gas. So that's why carbon cycle is considered as the gaseous cycle. Nitrogen is also considered as the gaseous cycle because ammonia, then nitrite, nitrate, these are the gases, and these gases are present in the uh, atmosphere. So again, the nitrogen cycle is also considered as the gaseous. But if we are talking about the phosphorus, then this is mostly phosphorus is not present in the atmosphere. It is present in the rocks and sediments. So that's why this phosphorus lacks a significant gaseous phase and is mainly stored in rocks and sediments. So C is the right answer. And just see here, I tried to give the explanation. So in the left hand side we have phosphorus cycle, and in the right side we have the carbon cycle. And just see here, we have carbon dioxide and we have respiration. So everything, so there is the presence of this carbon in the atmosphere, right? But here if we see the phosphorus, then this phosphorus is in soil, this phosphorus is in decomposer, in plants, in geological uplifts, and in sediments. So it is not in the gaseous form, right? So that's why it is termed as sedimentary. Just see the writer, because it does not include a significant atmospheric component. Instead, phosphorus is mainly stored in terrestrial rock and sediments, entering ecosystem through processes like withering. This contrasts with the carbon cycle, which have the substantial atmospheric phase involving CO2. So that's why uh, we consider phosphorus cycle as the sedimentary cycle. Coming to the next question, uh, in which of the following way does the carbon cycle differ from the phosphorus cycle regarding their primary reservoir? From that cycle, we already have learned, but right now you have to give me the answer. Based on the reservoir, you have to tell me the difference between carbon cycle and the phosphorus cycle. So we have four options here. Carbon is primarily stored in the earth crust, while phosphorus is mainly in the atmosphere. This carbon main reservoir is the atmosphere, while phosphorus is mainly stored into rocks and sediments. C is both carbon and phosphorus are mainly stored in the atmosphere, and D is both carbon and phosphorus are primarily stored in living organisms. What do you think? What is the right answer? What is the main reservoir for the phosphorus and what is the main reservoir for the carbon? We already have learned from that phosphorus and carbon cycle that I showed you in the last slide. From that, you can have some idea. So the answer is carbon main source, main reservoir is in an atmosphere, but phosphorus is mainly stored in the rocks and sediments. So the primary reservoir for the carbon is atmosphere, but the primary reservoir for the phosphorus is rocks and sediments. We already have learned it, right? So again, we have the same figure, and as I told you, the carbon cycle include a significant atmospheric component, with CO2 being a major reservoir. In contrast, phosphorus cycle lacks a gaseous phase, with its primary reservoir being terrestrial rock and sediments, and this is the fundamental difference that affects how each element move through the environment. Okay, so I'm going to the next question. Now, again, you have to tell me the difference between carbon and phosphorus cycle, but here in term of their movement through ecosystem. So how they move. So we have four options again. Carbon and phosphorus both cycle rapidly through the atmosphere. B is phosphorus cycles quickly due to its gaseous phase, unlike carbon. C is carbon cycle through the atmosphere, ocean, and living organism, while phosphorus moves more slowly through soil and sediments. D is phosphorus cycle rapidly through aquatic system similar to carbon. So in terms of their movement through ecosystem, how this phosphorus and carbon moves in the ecosystem, there is a difference between these two cycle, and you have to tell the difference between these two cycles based on their movement. And if you know the carbon cycle and the phosphorus cycle, I think you can give the answer for this. So we have learned that carbon cycle where the CO2 moves through atmosphere, ocean and living organism. But if we are talking about the phosphorus, this is sedimentary cycle, right? It is mostly present in rocks and sediments. So it moves more slowly through soils and sediments because this is not gaseous in nature, right? So that's why C answer is the right one. So just see here, the carbon cycle is dynamic with carbon moving swiftly between atmosphere, ocean and living organism. In contrast, the phosphorus cycle lacks an atmospheric component and proceeds more slowly, primarily through soil, rocks and sediments. And this results in slower turnover rate for phosphorus in ecosystem. If there is any doubt, you can write in the chat box. I will try to clarify, but if you do not have, then I will move on to the next question. Okay. Another question is, because we are talking about nitrogen cycle, we are talking about the carbon cycle, we are talking about the sulfur cycle, iron cycle. But what is the significance of this biogeochemical cycle? This will come under biogeochemical cycle. Bio means it will move through the living organism. These chemicals. So either this chemical is nitrogen, or this chemical is oxygen, or this carbon is carbon. So this chemical will move to humans or to the biotic or to the living organism, and even to the geo is a non-living form or sediments or rocks. So your chemical will move in the biotic as well as in the abiotic components. So this is how there is a biogeochemical cycle. But what is the significance? Why we are studying nitrogen, oxygen, carbon, sulfur, iron cycles in the ecosystem? So we have four options. They facilitate the one-way flow of energy from producer to consumer. B option is they enable the recycling of essential element between living organism and the environment. C option is they prevent the movement of nutrients between different part of the earth. And D is they are responsible for the production of energy within organism. So, like, why we are studying this different type of cycles? What is the significance of studying the biogeochemical cycles? And because this is cycle, from that only, like, there is a hint. If it is a cycle, cycle means your chemicals are being recycled, right? So we are having oxygen, and we are taking oxygen, right? And then CO2 will be produced. Then again there are something that will take the CO2 and will release the oxygen. So there is the recycling of the essential element between living organism and the environment. So that's why these biogeochemical cycles are very important to study. So B is the right answer. They enable the recycling of essential elements between a living organism and environment. Let me show you the explanation for this. I'm not able to clear this. Okay, so biogeochemical cycle, also known as the nutrient cycle, describe the movement and transformation of essential elements like carbon, nitrogen, phosphorus through the atmosphere, through the lithosphere, through the hydrosphere and biosphere. And these cycles ensure that nutrients are recycled and made available for use by living organisms that will maintain the ecosystem stability and function. Okay, so coming to the next question. The next question is, which of the following statement accurately differentiate energy flow from nutrient cycling in the ecosystem? So we have learned we have nitrogen cycle, we have carbon cycle, let's say. But how these cycle? Because these are the nutrients, that nutrients we need, and these nutrients are recycled. But how this nutrient cycle is different from the energy flow? So we have four options. Energy flow is cyclic, while nutrient cycling is unidirectional. B option is both energy and nutrient flow unidirectly through ecosystem. C is energy flow is unidirectional, where nutrient cycling is cyclic. D is neither energy nor nutrients are recycled in ecosystem. So how this recycling or the cycling of the nutrient, let's say nitrogen, carbon, how this is different from the energy flow? Because here the energy flow is unidirectional. So for example, if we get energy from the food, and and if someone will eat us, then some of the energy will waste in the form of the heat. So this is not like that energy will be utilized by the plant. No, that energy will not be utilized by the plant. Energy will be transformed into another energy, and this is only one directional. But if there will be recycling of the nutrients. So for example, we are taking carbon from the plants, and then after we die, or after the burning of the human, then again carbon will be released into the atmosphere, and this carbon will be taken up by the plants. So the nutrient cyclic is cyclic in nature. But if we are talking about the energy flow, then this is unidirectional. The energy will not be taken up again by the plants. So this is unidirectional. So just see here, in ecosystem, energy flow in a one-way stream from primary producer to various consumer and is eventually lost as heat. But in contrast, nutrients are recycled within the ecosystem through biogeochemical cycle, allowing for the continuous reuse of essential elements by living organisms. So this is how we can differentiate between the nitrogen nutrient cycling and the energy flow. Energy flow will always be unidirectional, and nutrient is cycling means we are like utilizing it again and again. So this is cyclic in nature. Coming to the next question, we have two kind of bacterias, nitromonas and nitrobacter, and these two bacterias are utilized in the nitrogen cycle. So you have to tell me what kind of reaction they perform in the nitrogen cycle. And we have four options here. Nitrosomonas convert ammonia to nitrate, nitrobacter convert nitrate to nitrogen gas. B option is nitromonas convert nitrite to nitrate, and nitrobacter will convert nitrate to ammonia. C option is nitromonas oxidize ammonia to nitrite, nitrobacter oxidize nitrite to nitrate. D is nitromonas and nitrobacter both convert nitrate to nitrogen gas. So what is the role of two kind of bacteria that we have, nitrosomonas and nitrobacter, in the nitrogen cycle? So we have learned, like in the atmosphere we have N2. This N2 will change into NH3. This will change into NO2, and this will change into NO3. And this is minus, and this NO3 will again changed into N2. So this is nitrogen cycle, and you just have to place bacteria in between this mechanism. So if we are talking about the nitrobacter, then nitrobacter will work here. If we have ammonia, sorry, I gave you the wrong explanation. Nitrosomonas will be used here and nitrobacter will be used here. So this is wrong. This is wrong. So just see here, if we do have nitromonas, then it will change from ammonia to nitrite. And if we have nitrobacter bacteria, then that bacteria will change nitrite to nitrate. And based on that, we have a C option, that nitromonas will oxidize ammonia to nitrate and nitrobacter oxidized nitrite to nitrate. So C is the right option. Is it clear? And I don't think that I should go to the explanation because we already have talked much about this. Okay, the next question is, why is the activity of nitromonas and nitrobacter bacter crucial for the plant nutrition in ecosystem? And because we already have talked, right, nitromonas will convert ammonia to nitrite and nitrobacter will convert this nitrite to nitrate. And that's why what they will do, they will transform ammonia to nitrate, and because this nitrate, whatever will be produced after nitromonas and nitrobacter, this will be taken up by the plants. Okay, so that's why D is the right answer for this question. I don't think that I should go to the explanation. And okay, you have to tell me the environmental condition. Under that condition, nitromonas and nitrobacter can thrive or can survive in the nitrogen cycle. Because as I told you, nitromonas and nitrobacter will change ammonia to nitrate. So ammonia is NH3. We do not have oxygen molecule here, and this nitromonas and nitrobacter will change this NH3 to NO2 and NO3. So now oxygen has been added up. So it means the condition should be aerobic. right? So if there is a aerobic condition and if we do have sufficient oxygen supply, then only this nitromonas and nitrobacteria will work. So that's why B answer is the right one. Okay, coming to the next question, we have another type of bacteria, and that bacteria is, you must have learned, there is a thiobacillus bacteria, and right now the name of this thiobacillus bacteria to acidothiobacillus thiooxidans. So right now the name is not thiobacillus, but it is acidithiobacillus. Don't confuse with the terminology. So you just need to tell me the metabolic role of this kind of bacteria in the sulfur cycle. Like different bacteria have different role, because they thrive in different condition, and the metabolism is very different from one bacteria to another bacteria. So if we are talking about the metabolic role of this bacteria, thiobacillus thiooxidans, then it will oxidize your elemental sulfur and thiosulfate to sulfate, and this is how it can produce the sulfuric acid. And that's why in the mining area, if you have such kind of bacteria, and in the mining area if we have plenty of elemental sulfur, then there will be the uh, sulfuric acid in, and this will create the acid mine drainage. So this is the role of that particular bacteria. I don't think that I should go to. You can read. I will share this slide, and anytime you can visit these slides. Yeah, okay. The next is what kind of environment is needed for this bacteria, and here you can see it is acid loving bacteria, right? So if there is the acidic environment, then it will be thrive, and this is used in sulfur cycle, so there must be sulfur compound here. So in that condition, this kind of bacteria can thrive. I should skip this. Then how does the availability of this particular bacteria impact the availability of sulfur for plant uptake? Because it will oxidize to sulfate, we already have learned, and this is how it will enhance the sulfur availability for the plant. Plant also need the element, for example sulfur, nitrogen, carbon. So if such kind of bacteria are changing elemental sulfur to sulfate, then this sulfate will be utilized by the plants. Then I think we already have talked much about the nitrogen cycle. Then let me skip this. You can see these slides. This is again the question on nitrogen cycle only, and we have learned much. Okay, the next question is about the role of fermentation, because like in microbiology, there is the application of microbiology in industries where we do a lot of fermentation. So what can best describe the role of fermentation in industrial microbiology? So here, after that fermentation, most of the time the end product that will be will be antibiotic and vitamins, and these are used in the pharmaceutical world. So that's why in industry, in the like microbiology, industrial microbiology, the role of fermentation is that it will produce the end product like antibiotics and vitamins that we need. And that's why this the scope of industrial microbiology is very vast. Coming to the next question. Okay, so like in industrial fermentation, we have primary metabolites and the secondary metabolites. If it is a primary metabolites, then it will be citric acid. But if we do have secondary metabolite, I think next question. If we do have the secondary metabolite, then secondary metabolites uh, are produced during the stationary phase and are not essential for the growth. So the remaining options that we had will come under the secondary metabolites. I'm just skipping the question because the slides will be available and you can see that slide later on. But I just want to take important question for next 2 minute, and then we will be like finishing this session. And then if we do have the sulfide ore, we can extract the sulfur from there. And what would be the reaction? So reaction will be, if we have the sulfide ore, this is the sulfide ore, we will use copper oxide, and this is how we can extract the copper. So C answer is the right answer. Coming to the next question, I'm just skipping everything very quickly. Here, if we want to extract the copper from the sulfide ore, then which gas is released as the byproduct? Then, of course, this is sulfur dioxide that will be released. You can just read the, and these are more questions on how we can extract the copper from sulfur or sulfide ore. And here copper sulfide will act as the reducing agent that will convert copper oxide to metallic copper. And there is another problem that we have, because we talked about methyl mercury is very toxic foam. And why this is toxic foam? Because that particular compound, methyl mercury, will bind to the sh bond that we have protein, and this is how it can degrade the protein that we have in human, and that's how it will impair the neurological function of the human. So that's why B is the right answer. So just you can see here, you can see the explanation. Then why fetus are very vulnerable to methyl mercury exposure? Because this methyl mercury can easily cross the placental barrier, and that's how it can affect the developing nervous system in the fetus. So this methyl mercury, that's why it's very toxic to the human. So C will be the right answer. Then if you will see the distribution of carbon in the earth, then you will get to know that more of the carbon is present in the deep ocean. And just here see that it is like the write-up is saying that 37,000 billion ton of carbon is present in the ocean, and this is, I think, little overestimation. So and just see the comparison with the terrestrial surface and the fossil fuel. So how much more billion ton of carbon we have in the ocean? So the largest reservoir of the carbon is the ocean. And I think I should stop here because time has up. And let me go to the next slide. I should end. Okay, so the thing is, I also had a lot of question, but I think because we have time constraint, then my slides will be available to you online. So anytime you can either watch the video or you can access the slide from NPTEL platform only. So thank you so much. Thank you so much for showing your patience. And we will have last summary class where I will try to cover all the content that we have covered from week 1 to week 12, and that live session will be day after tomorrow. So it means it will be on 16th April, and the timing will be same, 6 to 8 p.m. And thank you so much, guys. If you have any query, any suggestion, anytime you can tell me. My email ID is jyoti, JYOTI 21 @icb.ac.in. If you have any query related to like any topic, or if you want to learn more, you anytime you can like mail me. I will try to answer you as soon as possible. Thank you so much. Thank you so much. Okay, I got thank you response from students. So thank you so much, guys. Like, you are fabulous. Thank you.